Hello everyone. So, welcome to this lecture 44 of week 9. This week we have started discussing about the advanced wastewater treatment systems, or which typically we call tertiary treatment systems, because they are usually employed after secondary treatment systems. So, we call them as a tertiary treatment systems or advanced treatment system. In earlier lecture, the first of this week, we did discuss about what are the various issues with the existing conventional treatment plants which treat up to the secondary stage, and what are the contaminants that are still there in the water, and what kind of targets is set for the advanced treatment system. So, basically what do we need to remove as a tertiary treatment. So, one of the thing was nutrient, which is typically not removed that well in a conventional treatment systems, which usually leads more than like particularly for the domestic sewage, it usually leaves more than 20 milligram per liter of TK and more than say 1 milligram per liter of phosphorus. So, in order to remove the nutrients, we have to if particularly depending on the criteria, as we discussed, that for waters which is might be going to use for agricultural purpose, we may not need to bother that much about the nutrient removal, but particularly the one which is going to be disposed in say some water body, the nutrient pollution could be of point of concern or could be a point of botheration from that perspective. So, one of the things that is dealt in the tertiary treatment systems or advanced treatment systems is the nutrient removal, which we are going to discuss in this lecture. So, excessive nutrients like nitrogen and phosphorus, normally that is what we refer as nutrients. So, these disrupt the natural environment. Okay. The pollution in the surface water bodies kind of fuels the growth of harmful algae, which is typically known as algal bloom, or the more common word eutrophication. Okay. And this eutrophication have the ability of completely devastating the aquatic ecosystem. Okay. It is pretty common. I am sure majority of you must have seen some water body which is completely covered by the green top. So, that is actually a eutrophic water body, and that is what is called eutrophication. So, this eutrophication is caused by the nutrients, and when kind of nutrient pollutions are there, it may create the dead zones, which is also called hypoxia. So, this is where aquatic life cannot survive, okay, and they become dead, and this primarily happens because very little or no oxygen. So, when this eutrophication happens, the kind of oxygen will be consumed by these algae, and furthermore the rate of oxygen transfer also decreased, because oxygen transferred to this air water interface, and since that water top of the water is covered by this algae, by this kind of green material. So, the rate at which oxygen transfer to the water body, to the kind of lower portion of the water body, also also gets quite slow, and as a result not enough oxygen goes to the water, and whatsoever is available is consumed by the algae. So, that is why we get very little DO or almost no oxygen in the water which is covered by the green top. So, that can create the dead zone, which is called hypoxia, and the aquatic life cannot survive there. Okay. Water may have higher than acceptable kind of nitrate levels also. So, if somebody is, let us say, willing to use this water for some purpose, so the level of nitrate could be higher in these things. Typically 10 milligram per liter is permitted, but it could be much higher than that, which can basically restrict the usage of this for substantial purposes, and particularly for the domestic application or drinking water purposes. Apart from this, algal bloom can create kind of toxins which can kill the fish and other animals in the water. So, DO depletion or DO decrease is one aspect, but it can actually lead to add some toxicants as well. Okay. So, if these toxicants are consumed by the small fish, they will become dead. Then through this food chain, like these are consumed by the bigger fishes, and then kind of other animals in the water, and from there it can actually reach to even the human food chain. So, even if algal blooms are kind of not toxic, they can hurt the aquatic life by blocking out the sunlight, clogging the fish gill. So, there are variety of these issues, and eutrophication is one of the most sought after environmental problem across the globe, across the world. And the primary reason for eutrophication is the nutrient, which is usually disposed of either through agricultural runoff to the natural water bodies. Agricultural runoff is one of the major sources of the nutrient, okay, because lot of fertilizers are used in the agricultural activity. Nitrogen and phosphorus compounds are there in the soil, and whenever there is any rainfall happens, so through this agricultural runoff, these leads to the nearby water bodies and make them eutrophic. Whereas the discharge from the like sewage is also one of the point of concerns for the eutrophication point of view. So, nutrient removal that way is an important process in meeting high quality effluent requirement, particularly if it is being targeted for discharge. So, how do we remove nutrients? Nutrients, basically we will talk about the removal of nitrogen and phosphorus throughout this lecture, and then removal of nitrogen and phosphorus both can be achieved through chemical or physicochemical means as well as through biological means. Okay. So, nitrogen removal, if we see, there are the one of the most common process is nitrification, denitrification, which is a biological method for nitrogen removal. There is NMOX, which is also kind of is being more popular these days. Then there is ammonia stripping, which is physicochemical method. Apart from that, there are a couple of other methods which are there, but very rarely used, like the breakpoint chlorination, where adding the chlorine actually kind of first converts ammonia to the various chloramines, monochloramine, dichloramine, trichloramine, and then it can eventually break that chloramine as well to release the nitrogen. So, that is one way. Then of course, there are, because nitrates or ammonia are in the form of ions, so ion exchange kind of systems can be used, which we will discuss later in this week, but not from the nutrient removal perspective, because it is very rarely used for nutrient removal. Okay. And of course, the membrane processes can remove various dissolved ions, so including nitrate, nitrite or ammonia, those kind of things can also be removed by those, but they are high end treatment processes and generally not deployed for removal of the nutrients or nitrogen that way. Then for phosphorus removal, we have biological phosphorus removal and chemical phosphorus removal methods, which we will be discussing. Typical phosphorus concentration in domestic sewage, what we get is 4 to 8 milligram per liter, whereas the nitrogen concentration, total nitrogen concentration is 25 to 50, out of which 15 to 35 remains as ammonia and 10 to 20 as organic nitrogen. Organic nitrogen also can be kind of through hydrolysis can be solubilized in the form of ammonia. So, the major target for the removal nitrogen removal is the removal of ammonia. Okay, that is the one thing in which, like, nitrogen usually remains dissolved in the state of ammonia until unless it actually is oxidized that way. Okay. Some nitrite and nitrate could also be there. So, these are the kind of things that we will be discussing. Okay. Then if we see, so the nitrogen removal, particularly the nitrification or denitrification process, is essentially a biological process. So, the bacteria removes nitrogen from the wastewater in this two-stage process, which is nitrification first, and that is followed by the denitrification. Okay. The conversion of ammonia to nitrate through this intermediate nitrite is called nitrification, whereas then reduction of this nitrate again through this nitrite eventually to the level of gas phase, okay, nitrogen gas, is called denitrification. Nitrification is kind of achieved by the autotrophic group of bacteria, whereas denitrification is by the facultative heterotrophic bacteria. Okay. And since this is an oxidation process usually, so this happens in the presence of oxygen, whereas the denitrification is takes place under anoxic conditions usually in the absence of oxygen. So, by providing additional aerobic biological treatment, these nitrifying bacteria, which are mostly present in the water anyway, can biologically convert ammonia to the non-toxic nitrate, which is the process is called nitrification, right. So, however, this needs larger residence time. Okay, so we need to give larger retention time if we want to achieve the nitrification process, because generally the nitrifiers are slow growers, so they grow slow and they act upon slow. So, in the conventional treatment system, the residence time that we provide is not good enough for nitrogen, so there is not much of nitrogen removal takes place, and only it is the CBOD or the carbonaceous BOD which eventually gets removed in the conventional activated sludge process or those conventional aerobic treatment systems. So, if we intend to basically remove the nitrogen or particularly the ammonia, so what we can do, we can go for a two-step nitrification process, okay, which is kind of which therefore which works with a nitrosomonas bacteria that converts ammonia or ammonium into the nitrate, and then there is a nitrobacter that converts nitrate to nitrite. So, this reactions is coupled and kind of proceeds rapidly to the form of nitrate. So, that is why the nitrite level at any time, because as soon as the nitrosomonas convert ammonia or ammonium into nitrite, the nitrobacter immediately converts that nitrite to nitrate. So, that is why the level of nitrite is always very low in the system, which is good also because that is a toxic species, whereas nitrate is far less toxic. The nitrification process is normally sufficient to remove the toxicity associated with the ammonia in the effluent, and so for several purposes where we are just bothered about the toxicity, we may not even need to go for any additional step, just nitrification is good enough because ammonia has been converted to nitrate and then we release it off, or we reuse at a nutrient because nitrate fertilizers or those kind of thing. However, in cases where we need to remove the nitrogen completely from the system, we must go for an additional biological process which converts nitrate to nitrogen gas, or is basically referred as denitrification. Okay. And this is done in an anoxic environment, and bacteria use basically the oxygen which is attached, because it is an anoxic environment, there is or anaerobic environment, there is no oxygen present in the system. So, bacteria use the oxygen attached to the nitrogen in the form of nitrate, okay, and this nitrate becomes the terminal electron acceptor in this case, and in the presence of a carbon source, they kind of in the process of degradation of carbon source, they consume that, they in order to get that oxygen separate out from the nitrogen, they release the nitrogen gas and assimilate or consume the oxygen present in the nitrate. So, the reaction is like this, we have nitrification, like the ammonia gets in the will react with the oxygen, will form nitrite, then this will further react with the oxygen, will form the nitrate nitrogen. So, overall reaction, the ammonia can be converted to the ammonia nitrogen can be converted to nitrate nitrogen. This dash N simply reflects that this is represented as nitrogen, we can remove that also. So, your reaction will still be same way even if we remove the N dash. Whereas denitrification process is again this nitrate nitrogen in the presence of carbon source and facultative bacteria, it converts to the nitrogen and form some new bacterial cell. So, if we consider the typical like we need a carbon source for this denitrification process. So, if we consider the typical carbon source available in the wastewater in the form of say C10H9 H19O3N. So, then this is the reaction where this nitrate reacts with this and eventually converts the nitrogen, converts it to nitrogen. Methanol can be another carbon source, or acetate can be another carbon source. So, any carbon source that way will be kind of able to, through denitrifiers, will be able to use that and convert the nitrate into the nitrogen. The effective nitrification depends on kind of sufficient oxygen and alkalinity that should be present in the system. Okay. So, typically this nitrifier requires 4.57 milligram of oxygen and 7.14 milligram of alkalinity as a calcium carbonate for each milligram of nitrate nitrogen formed, and they usually yield from 0.06 to 0.2 milligram per liter of biomass or VSS for each 1 milligram of nitrate nitrogen which is being formed, okay, in the process of nitrification. So, this typically requires a long residence time, a low food to microorganism ratio and a high mean cell residence time. That way the rate of nitrification can be kind of optimized by controlling these factors, and it eventually depends on the temperature and pH as well. So, like you can see here that as temperature increases the rate increases, pH again for like optimum we need a kind of optimum pH for this thing as well, and DO maintenance is also necessary for the achieving good growth rate of nitrifiers. These nitrifying organisms are present in almost all aerobic processes, okay, and that way, like under favorable conditions, when there is usually the BOD5 to TKN ratio is greater than 5, we can do this in the your typical activated sludge process itself. So, this carbon oxidation and nitrification we can actually do in a single reactor, although we need to give larger retention time there. Okay. So, that is called single stage or combined carbon oxidation and nitrification, or combined CBOD and NBOD removal, because we are removing carbonaceous BOD and nitrogen oxygen demand also together. So, since this is done in a single stage, so we call this a single stage process or combined oxidation and nitrification process, which can be accomplished in both suspended as well as attached growth systems, okay, like activated sludge process, rotating biological contractor, sequencing batch reactor, all those things. The nitrification could also be achieved in a separate reactor, which is called separate stage or two stage process. So, like we have a single stage process, we can go for a two stage process where the carbon oxidation and nitrification takes place in a separate stages and in a separate reactors. Okay. So, this typically for suspended growth systems, it's even the nitrification design is similar to the design of activated sludge process, whereas in attached growth system like trickling filter, rotating biological contractor or packed bed reactor could also be used for the separate stage nitrification. The separate stage nitrification is usually practiced when we have BOD 5 to TKN ratio usually less than 3. Okay. Now, so once nitrification is done, the denitrification is accomplished under anaerobic or near anaerobic conditions, where the facultative heterotrophic bacteria like Pseudomonas, Micrococcus or Archamobacter. So, all these bacillus, these species eventually they are available in the wastewater, typically they are present in the wastewater. So, these convert nitrate to either nitrogen gas by the bacterial metabolism or assimilate nitrogen into their cell mass. So, that is how the nitrogen is removed, the dissolved nitrate is removed from the system. This denitrification occurs when the oxygen levels are depleted and the nitrogen or the nitrate which is there becomes the primary electron acceptor. Okay. So, if there is oxygen, so in the process that actually accepts electron, but in the absence of oxygen nitrate becomes the electron acceptor, and that way it is reduced further by accepting electrons. These denitrifiers require organic carbon for energy, as we were just seeing that it can use the organic carbon present in the wastewater itself. So, if nothing is present, the endogenous decay of the organisms could itself provide source of organic carbon. However, for faster rate it is preferred that there should be some carbon source present in the seepage. in the wastewater. So, if the carbon source is present in the wastewater, it is fine. If not, a supplemental source organic carbon like methanol or acetate, those kind of things can be supplemented for the process for the organic carbon requirements of these anaerobes. So, these are a couple of these things. So, we can have a suspended growth separate stage nitrification process, where this is kind of a separate stage nitrification process. So, we see that sludge is coming, first it is going to the aeration level for BOD removal, secondary clarifier is happening, and then there is another aeration basin for nitrification, and then there is a clarifier for nitrate. So, we are actually getting two stages of sludge production as well. This is a typical suspended growth system. We can have single stage nitrification as well. So, we get rid of the other part, and the BOD removal and nitrification is done in a single aeration basin. Only thing is that residence time has to be given little longer here. Okay. So, there are a couple of pictures which has not come somehow, but you will get that in the materials when it is uploaded anyway. There could be various different configurations like pre-anoxic denitrification. So, in this one what is done that we for denitrification purpose we put an anoxic zone before the nitrification. So, whatsoever is there, it is converted first, then it is again converted to nitrate, and then nitrate feed is recycled back to the aerobic system, so that nitrogen is removed. So, that is when we add prior hand, we can have kind of post anoxic system as well. So, where the anoxic zone is kept after aerobic. So, there is no need of returning nitrate feed over here. Similarly, we can have single stage system or two stage sludge production system as just we were seeing. So, we can have a like nitrification in a separate this thing and denitrification in a separate system. Okay, so that is two stage sludge systems, and here since denitrification and nitrification are taking place in this thing, and then it is going to secondary clarifier. So, sludge which is generated contains both kind of sludge, whereas here these nitrifier and denitrifier sludge is kind of separate. So, we have that way two stage denitrification can also happen. There is another biological nitrogen removal process which is NMOX, which is kind of a newer term or newer technology. So, this is NMOX is essentially anaerobic ammonium oxidation, which we typically call as NMOX. Okay. So, this is discovered in early 90s, and what it does, so instead of going for the complete process of nitrification and denitrification, it converts the ammonium and nitrite through this partial nitrification into the nitrogen directly. So, we get rid of the conversion into the nitrate and then denitrification of nitrate with a source of acetate. All these things are actually we get away with this. So, just ammonium ion and nitrite ion are converted to nitrate. It actually the NMOX bacteria, which is kind of Candidatus brocadica NMOX. So, that actually is used here, and this reaction is something like this. So, ammonia will react with the nitrite and form nitrogen and H2O. This process has several distinct advantages. Okay. The oxygen addition can be reduced because we are not going for complete nitrification, we are just going for partial nitrification. Then these bacteria do not require organic carbon source like methanol and those kind of thing, we are not going for denitrification anyway. So, production of sludge is also reduced because larger aeration, larger biomass reduction, and the reduction of CO2 is also kind of is there. CO2 emission is decreased. So, these are the advantages, whereas it is a process under development. So, there is kind of a lot of research still needed, and there is a high construction cost of these processes. So, these are kind of the demerits of this. Now, if we compare it with conventional processes, so in conventional processes we have ammonia coming through a nitrification basin and then converting into the nitrate, which is going to the denitrification, releasing nitrogen and effluent, and in the process it needs a methanol or some organic carbon source, whereas in NMOX process it is just partial nitrification and then the remaining ammonia and nitrate reacts and kind of forms N2 that way, okay, and little NO3 which goes into the effluent. So, that way is the NMOX process is used. If we compare your typical these steps are gone here, as you can see here in NMOX process. The reactor volume becomes small, the oxygen requirements become small, we do not need any further chemical dosing like methanol or those kind of thing, sludge production is small, okay, and kind of optimum temperature, this works at 25 to 37 degrees Celsius. So, temperature control is essential, it cannot work in very cold climate that way. There is air or ammonia stripping, which is another method for removal of the nitrogen. So, the ammonia stripping is again a physicochemical process, not a biological process that way. So, what happens that because there are kind of ammonia present in the system, and ammonia is ammonium ion particularly is such a species that they remain gaseous ammonia and aqueous phase ammonium ion remains together in equilibrium, and their equilibrium is controlled by the pH and temperature. So, depending on the pH, what pH is and what is the kind of temperature, as we see the different temperatures, we can see what fraction of ammonia or what fraction of ammonium ion will be there in the water. So, just by controlling the pH, particularly if we move the pH towards alkaline zone, say around 12 or 11, if we are able to make pH around 11 or 12. So, we see that the maximum portion will be actually in the form of ammonia gas and not in the ammonium ion. So, ammonium ion is something which is dissolved in the water, but if it is converted to the ammonia gas, okay, which predominantly exists like kind of unionized gaseous form. So, that can be kind of then released back, okay. So, this can be made to escape to the atmosphere if we go for a air stripping process, okay. So, the idea here is that dissolved ammonium is converted to the gaseous phase and then dispersed in the air. This allows kind of transfer of the ammonia from wastewater to the air, and pH must be greater than 11 for complete conversion of ammonia. So, around greater than 11 pH we see at around 20 degrees Celsius we have here 99% of 98 to 99% of actually the total nitrogen remaining in the form of ammonia and not the ammonium ion. Ammonium ion is soluble in water, that will remain in the water, but if it is converted to ammonia gas, so this gas can be basically then stripped off, okay, which is done in the ammonia stripping process. So, typically kind of lime is the most common means for raising the pH. Lime is added so that pH is raised. There is enough lime must be added to precipitate the alkalinity and to kind of increase the OH ion for the pH adjustment. The air to water ratio ranges from 2000 to 6000 meter cube of air per meter cube of wastewater, which is typically used in the design, and this works well for 100 to 10 to 100 milligram per liter range, okay. There are different kind of strippers which can be used. There are packed bed strippers when there is a packing of this thing. So, we kind of flow liquid from the top and air from the bottom. So, as they pass they interact with each other, air gets out, liquid gets collected in the bottom, or we can have a sieve tray. So, where there are trays, again liquid moves in. So, liquid actually falls from these trays to each other, and then there is a air which actually kind of through these pores and bubbles moves up. So, that kind of aerator is there. This is how typically a stripping system looks like. Another nutrient that is there in the water is phosphorus, which kind of normally secondary treatment can remove 1 to 2 milligram of phosphorus. So, a large excess of phosphorus still remains in the outlet of the secondary unit. There is kind of different forms of phosphorus. It could be in the form of orthophosphates, polyphosphates, or originally bound phosphorus. This removal of phosphorus can be achieved through chemical methods or biological methods. Chemical methods are more reliable and easy to operate, that is why more popular, although they are expensive and cause of kind of increased sludge volume which could increase as high as around 40%. The alternate biological phosphorus removal methods are also there. Okay, they are kind of more environmental friendly, which are achieved by sequencing and producing the appropriate environmental conditions in the reactor where the bacteria assimilates the phosphorus. So, from the biological methods there are specific bacteria which is poly-P heterotrophic bacteria. Okay. This is also called polyphosphate accumulating organisms or PAOs. So, they have potential to accumulate large quantities of phosphorus within their cell mass. So, in their cell structure they can accumulate as high as around 20% of the total mass could be actually of the phosphorus. Okay. And but that is not in the usual case, that is under specific growth regime, and we have to provide that growth regime. That growth regime is kind of ensured by a anaerobic aerobic sequencing. The COD to phosphorus ratio should be greater than 45 and there has to be high phosphorus level in the system. So, for effective removal these microorganisms PAOs are transferred back and forth between the controlled environment that first forces them to release the phosphorus and then they first release the phosphorus from their cell, and then once they again introduced in the other system they absorb more phosphorus that they would normally have, and that is why the accumulation of phosphorus increases. When this biomass enriched in these bacteria is separated from the treated water, the phosphorus level in the water will be decreased because most of the majority of the phosphorus has been assimilated into the cell mass. Okay. And the cell mass which is separated because of its high phosphorus content has a very high fertilizer value. So, these biosolids are helpful in that way as well. So, there is kind of it is, as we said, that it is an anaerobic aerobic sequence process. So, what happens that there has to be presence of sufficient VFAs in the anaerobic reactor upstream, and which kind of from where the flow goes to the aerobic system. So, this VFAs helps in development of these bacteria PAOs, okay, and that because in conventional activated system they will not develop that much. So, this anoxic environments helps in that development of those microorganisms. So, what happens that in the anaerobic environment, if we see in the anaerobic environment these the PAOs absorb these VFAs which are there, okay, in their cells, and in the form of kind of internal polymer such as PHBs. So, these VFAs are absorbed as a PHBs in their cell in the anaerobic this thing, okay, and the phosphorus, that polyphosphorus that we have, okay, that is also kind of converted, is used as a source of energy. So, the ATP is generated which is used as a energy, and in the process this phosphate is released from the cell. This happens in the anaerobic environment. Then when we move to the sequential aerobic system, so in there these PHBs is which are kind of assimilated VFAs, and then these PHBs are used to form the new cell and to generate the energy to fulfill the energy requirement as well. So, through this NADH they produce ATP and they produce new cell mass as well. Okay. So, part of this release energy which is released is again utilized to regenerate the polyphosphate in the cell, and in the process this phosphate which has been released in the which is there in the water gets kind of assimilated, gets consumed. So, in this process phosphate is absorbed from the liquid phase by these PAOs, and that way the concentration of phosphorus decreases and it actually moves to the cell. So, there are kind of there are mainstream processes where we can have anoxic reactor followed by an kind of aerobic system right away, or we can have a side stream process where this anaerobic process done is in a side stream and then actually it is again supplied to the conventional aerobic system that way. So, then there are a couple of chemical approaches for phosphorus removal as well. So, the biological approach was the one that we discussed. Chemical approach, it is actually simple chemical precipitation. So, either lime based precipitation, okay, or alum based kind of flocculation and precipitation is what is used. So, lime based precipitation, there is excess lime is added in the wastewater, and which kind of cause phosphorus to form precipitate with the calcium hydroxide. This precipitate then can be flocculated and removed from the water by simple settling. So, what happens, that calcium is there, it will react with the phosphate and it will form this hydroxylapatite, which is kind of precipitate. So, this is heavier, this will settle down in the system. The amount of lime required for this treatment is generally independent of the amount of phosphorus present in the system, okay, and will basically depend on the alkalinity, because there has to be adequate alkalinity present in the system for this reaction to take place. Typically pH of the wastewater will be raised because we are adding lime. So, pH will be raised in the range of 10 to 11 with the addition of lime, okay, and that way the proper precipitation of phosphorus takes place, which is quite a reliable method. These slugs are heavier, so they quickly get settled out, whereas alum precipitation with the alum, so where aluminum sulphate, or what typically we call as alum, is added to the wastewater, and here a precipitate of this aluminum phosphate forms, okay. So, this alum reacts with the this thing, and a precipitate of this alum phosphate forms. This aluminum phosphate is not that a dense precipitate, and that is why it needs to be basically some kind of flocculation is needed, where we can add a polymer dose or something and it can flocculate that way. So, and then we can make it settle, make it precipitate, or we may actually need to go for the filtration as well, because of the presence of the microflocks of aluminum phosphate in the system, okay. So, when alum is added, it acts as an acid. This reduces the pH of the wastewater by reducing the alkalinity, because alkalinity will also be reduced. Alum will react with the alkalinity and form aluminium hydroxide, which also will precipitate. So, that way excess alum is reduced, but it kind of reduces the alkalinity as well. So, optimum phosphorus removal is generally achieved at a pH range of approximately 6 to 7, okay. Apart from these, there is kind of iron salts, kind of ferric chloride, or those kind of coagulant or can also be used for the removal of phosphorus, okay. However, these are the more popular ones. So, that way the chemical removal of the phosphorus takes place, okay. So, with this we kind of conclude this session here, okay, and in the next class we will take up the removal of we will take up some other advanced treatment approaches or tertiary treatment approaches for the removal of the remaining contaminants. So, thank you and see you in the next class.